Good day, everyone. On behalf of Cambridge Health Tech Institute's Breakout COVID-19 Global Web Symposia Series and our sponsor, ACRO Biosystems, I'd like to welcome you to COVID-19 Serological Test Development. My name is Elizabeth Lamb, and I'm the host and moderator for today's event. Now, I'd like to introduce our presenters for today. First is Marco A. Biamonte, PhD, founder and CEO, Drugs and Diagnostics for Tropical Diseases. Our second presenter is Yinan Jiang, PhD, Product Development Manager with ACRO Biosystems. And our third presenter is Stephen A. Rawlings, MD, PhD, Clinical Fellow, Division of Infectious Diseases and Global Public Health at the University of California. So welcome, Dr. Biamonte. The presenter ball is yours.
Yeah, thank you very much. So yes, hello everyone. I wish I could see your beautiful faces, but I cannot. I'm very sorry about that. And again, reinstate my gratitude to Elizabeth Lamb and Cambridge Health Tech for to present, as well as to Larry Yu and ACRO Biosystems for the work they've done behind the scenes. So again, I'm the CEO of Drugs and Diagnostics for Tropical Diseases. The point today is not to showcase our organization, but to tell you about a product that we're developing, which is this universal IgG lateral flow assay, and we'll illustrate it in the context of COVID-19. If I manage to advance my slides, there we go. Just very briefly, again, for our organization, we're a not-for-profit organization. We're based in San Diego, California. And our mission is to develop new treatments and new diagnostics for diseases that affect disproportionately low-income populations. You can find more about us at ddtd.org. Very briefly, again, to come up with diagnostics for what the World Health Organization neglected tropical diseases. So it's a group of diseases that infect literally tens of millions of people and that are hardly heard of in the Western world. But here are three indications that we're working on. I have additional projects. So one is Loa Loa. It's a parasitic worm that can be seen sometimes traveling through the eye. There are 10 million cases in sub-Saharan Africa. Another disease is river blindness, also due to a worm where the larvae get into the eyes and lead to progressive blindness. There's 18 million cases. And a third disease we're working on is lymphatic filariasis, which is due to yet a third type of worm. And this time, the worms live in the lymphatic systems and they lead to these grossly enlarged limbs. So we're working normally for this type of diseases and this type of populations. And in the course of our work, we have developed a number of lateral flow assays. So lateral flow assays are these tests that look like pretty much a pregnancy test. Generally speaking, they represent a mature and cost-effective technology. They're really ideally suited to the point of care, to low resource environment. And one thing that for us is important, they're also heat stable, so you can work in tropical countries. This is how you would normally configure, or that's one possibility to configure a test to detect IgG antibodies, so a serological response to the spike protein, which, as you know, is one of the 4 proteins that make up the COVID-19, or rather the SARS-CoV-2 virus. So you would have a strip of nitrocellulose. You would immobilize a test line of recombinant spike protein. And then on the other hand, you would have some brightly colored nanoparticle that would be conjugated to a monoclonal antibody. And that antibody would be specific for human IgGs. So now, if in your sample you happen to have human IgGs that are specific for the spike protein, you form this sort of ternary complex. The human IgGs in your sample bind to the spike protein that's dispensed and immobilized on the nitrocellulose, and the brightly colored nanoparticles will recognize every IgG around and they will bind to it. And so you form, this is called a sandwich complex, and this leads to the accumulation of these colored nanoparticles at the test line. And therefore, you can have a visual readout even with the naked eye. The type of nanoparticles that are normally or frequently used are 40 nanometer gold nanoparticles. And here there's some interesting quantum physics phenomena that take place. When gold reaches the nanometer size, electron waves at the surface of the nanoparticle, and these electron waves are called plasmon, interact with the electromagnetic spectrum, and they strongly absorb visible light. And this leads to an intensely colored, actually very intensely colored material that is ideal to detect minute amounts of biological substances. So certain frequencies are absorbed more than others, and that leads to the characteristic red color. So it's gold, but it's deep red. Now, a couple of years ago, we introduced a novel class of plasmonic nanoparticles that were optimized for lateral assays. We introduced those with our with a company we've been working for many years, Nanocomposites, with a fantastic relationship. And we pioneered the use of what we call gold nanoshells to increase the sensitivity of lateral flow assays. So the gold nanoshells consist of a silica gel core, which is the gray ball that you can see here, that then gets studded with gold crystals, which are the little black dots that you can see here. And these gold crystals are used as nucleation sites to progressively grow a shell of gold, which is about 20 nanometers thick. The gold shell provides the optical properties, while the silica gel core really lowers the overall density of the nanoshells, and that allows them to be easily resuspended in water and to flow through the assay unimpeded. The benefits of these nanoshells is that they have even more weird surface plasmon resonance effects. Lead to a very dark blue color. It's nearly black. I like to call it blue gold. And here, each particle absorbs light 35 times, more efficiently than the regular 40 nanometer gold. So here's an example on this picture. On the left, you have the regular 40 nanometer gold. On the right, you have the gold nanoshells. On the left, you have 35 times more nanoparticles per milliliter than on the right. And yet on the right, it looks darker to the human eye. This leads to an increase in analytical sensitivity for these lateral flow assays of anywhere between 2 to 10-fold. So sometimes it's really minor, but sometimes actually quite important. And that depends on the exact assay. In addition, the black test line provides a high contrast readout that nice. So this is the result from a blinded test that was done at the NIH for one of the neglected tropical diseases I showed before, Loa, the worm that you can see in the eye. And it's pretty clear which patient has experienced or has not experienced that disease. Now, the gold nanoshells are not the only key to developing a sensitive assay. There's additional parameters that are importance to reach a good sensitivity. One is the nature of the anti-IgG antibody, so the monoclonal antibody, and chemistry that's used to conjugate that antibody to the nanoparticle. A second parameter are the blocking agents and the surfactants that are used, both within the test strips and also in the eluent that you add to the test. A third parameter is how the sample and nanoparticles are delivered to the test line. The timing matters a lot, actually. And even within the plastic housing, there are pressure points that regulate the flow of the various reagents. And we carefully design them to get really the optimal flow. So they don't look like much, but they play a critical role. And finally, there's another thing one needs to be mindful of, is that usually these are used with blood that's collected by finger prick. And so you need to have additional materials to retain the red blood cells and preventing them from migrating onto the assay. And for that, you need to add what's called a sample pad. But the sample pad, just the mere addition of that, can change completely the fluid dynamics. And as a result, blood does not necessarily behave as well as plasma. And I'll open a quick parenthesis. You often see tests like for COVID-19 characterized with plasma, but what you really want to see is to see them characterized with blood. So over the years, we've created this highly sensitive platform we have created a highly sensitive platform that's rapidly customizable to detect IgG antibodies that are specific for any antigen. So all the parameters have been optimized, but the test line, instead of being made of the spike protein, like I showed before, is made of streptavidin. And then the antigen, for instance, the spike protein, is biotinylated and it's dried within the test strip. So the spike biotinylated protein binds to the streptavidin test line, and if you will, the test line formed in situ. You put 10 microliters of sample in one port, 2 buffer in a second port, and you can read at 30 minutes. So this platform was originally developed for tropical diseases, I alluded to, but having this highly sensitive platform for us was natural to adapt it or retrofit it to COVID-19. And these are results that were obtained 2 months ago with a prototype that we had sent at the New York Blood Center. And you can see that most of the time you can see a clear test line. On the left-hand side, I purposely put the weakest test line that we saw, just so that I don't cheat and show you only the good results, but we've optimized the test since then. So where should you put that biotinylated antigen, like the biotinylated spike protein? Well, for a commercial product, that biotinylated antigen must be dried within the test strip. That's the easiest thing for the end user. They don't have to think about it. But for research purposes, you could think about putting the biotinylated antigen, mixing it directly with the sample and running that mixture as is, if you will, and that will allow you to get a quick sense for the assay performance. And we call that the universal IgG assay. So it's essentially a device that contains all the reagents except for the biotinylated antigen. All the parameters are optimized, so the amount of nanoparticles, the blocking agents, the buffer, the cassette, the sample pad, you name it, that represents years of work. And then the end user can just add the antigen in solution to the assay. And I'm going to illustrate that for SARS-CoV-2. So we will use positive control, a human IgG that was isolated from a patient, and then produced recombinantly from human cells. So this IgG is known to bind to the RBD domain of the spike-1 protein and is sold by ACRO Biosystems. By the way, I'm not getting any kickback for that. By ACRO Biosystems, and here is the catalog number. And we're going to check if this antibody is able to detect or bind to 2 different antigens, the biotinylated RBD domain of SARS-CoV-2, also available through ACRO, has a molecular weight of 36 kilodaltons, or the full S1 trimer, which includes the RBD domain, but has extra stuff around it, and which has a much higher molecular weight. So the procedure is the following one. We're going to prepare a contrived sample by diluting the recombinant IgG, negative human serum, or negative human blood. And then in parallel, we're going to prepare a stock solution of the biotinylated reagent with the above-mentioned IgG. I take that back. I had a moment of distraction. We want to prepare a stock solution of the biotinylated reagent, which the above-mentioned IgG should detect. And we're going to mix the contrived sample with the biotinylated reagent, place them together in the sample port, and then we're going to place again 2 drops of buffer and wait 30 minutes before doing a readout. And if the IgG binds to the antigen, as it should, then we'll see a positive readout at the streptavidin test line. So here's the first example. We're using as the biotinylated antigen the RBD domain, and the positive control, he's here at 10 micrograms per ml, 2 or even 1 microgram per ml, and here there is none. And we can clearly see a test line at 10, we can still see it at 2, and we can kind of see it at 1 microgram per ml. So we can detect as little as 1 microgram per ml of positive control, and I'll show that in a minute that this is really an excellent result. I should also add that these results were obtained in less than a day from start to finish. So again, the platform, as far as I am concerned, is actually very efficient. The second example is with the biotinylated S1 trimer. And again, we have the positive control, the IgG, at different concentrations. And again, we can easily see a test line at 10 and at 5 and at 2 and sort of at 1 microgram per ml. So the limit of detection is of the order of 1 microgram per ml of positive control. And again, that's very good. The universal test is so that you can use it with blood. And again, one would normally use, capillary blood. You would not dilute blood with an antigen. You can only do that in this universal platform, but we can easily see 10 and 5 micrograms per ml of antibody, and maybe 2 and 1 becomes really the limit of detection. So let's call the limit of detection in blood somewhere between 2 and 5 micrograms per ml for both the biotinylated S1 and the biotinylated RBD antigen. Here is what we use as a sort of comparator. We use a device that we do not have in hand, but that's made by Standard Diagnostics, and it's called the Duo Assay for COVID-19. Standard Diagnostics is a very serious company. It's a large company in South Korea. It's now part of Abbott. And if you look at their product insert, which by the way is really nicely made, it states that their limit of detection is of 20 micrograms per ml of IgG. Well, our limit of detection is more like 1 microgram per ml in plasma, or 2 to 5 micrograms in blood. And admittedly, we may not be using the same positive control or even matrix perhaps at Standard Diagnostics. So it's not fair to say that we are 20 times more sensitive, but I think it's at least fair to say that we do have a highly sensitive platform. So the benefits of the universal test is that this allows the user to rapidly adapt a new antigen to a new indication. And one can ask which antigen works best in a lateral flow assay for indication X versus limited to COVID-19. This gives groups without access to lateral flow assay manufacturing equipment or specific know-how the possibility to create a prototype in-house. You don't have to share confidential information, antigens, but you can use that to help management and funders to move to the next step. And our business plan is that we plan on start selling this test for research use only in about a month. You'll then be able to purchase it as you would any other reagent. You can apply it to any disease you wish. You're free to move forward as you like. There's no strings attached. You can choose. And if you want us to draw your antigen for you, we can do it. And if it is for global health purposes, basically if it's aligned with our mission, we will discount the service. And this is the last slide. I just would like to say that this universal platform meshes very well with the ACRO platform, because ACRO uses the AVITAG technology, where a sequence-specific enzyme is used to biotinylate one and only one lysine residue on the protein. And having a well-characterized biotinylated antigen is of definite interest for this platform. So this brings us to the end of the presentation. Again, I thank the organizers. You're more than welcome to shoot us an email, either to myself or to Melissa Chalmers, our Executive Directors of Development. We're always happy to hear from you. And Elizabeth, I will stop here and pass the mic.
All right, thank you. We do have a question that has come in from Falk. How about other Ig classes such as MDEA and incomplete antibodies?
So we don't have that yet. What we have essentially finished as well is to detect the IgG4 subclass. IgG4s are often expressed or overexpressed in parasitic diseases, which is what our area of expertise. We have not done other antibody classes other than these two.
Thank you. And one last question. This one is from Israel. He works in a third-party lab, and they've received many requests from clients who want to register an IgG or IgM kit for COVID-19. We propose to evaluate sensibility, the use of plasma and serum obtained from patients with a nasopharyngeal PCR positive test to COVID-19. But for specificity, they need to test it against different antibodies for other diseases. Do you have any recommendation of which antibodies to select? We think we can use standardized purchase antibodies against influenza A, B, and MERS.
So what I would recommend is to this person is that, so I understand that this person is in Israel, but FDA has guidelines and a template on what they want to see from serological manufacturers when they validate lateral flow assays. And I would refer the person to that document. I'm not sure that MERS is that important, but for instance, the FDA wants to see at least 10 HIV samples being evaluated. I hope that answers the question.
All right. Thank you so much, Dr. Biamonte. Now we will get ready to hear from our second presenter for today. Yinan was not able to be with us due to the time difference. So what we will be doing, he did record his presentation for us, and we will be playing that back for you momentarily. So here is Dr. Jiang.
Hi everyone, this is Yinan from ACRO Biosystems. Thank you very much for having me here. It's a great honor for me to have this opportunity to share with you the critical role of antigens in COVID-19 serological test development. In addition, I would like to talk about what ACRO Biosystems has done to help fight against COVID-19. First, I will give you a very brief introduction of myself. My name is Yinan Jiang. I obtained a PhD degree in structural biology from Tsinghua University. My PhD studies focused on elucidating the molecular mechanism of viral cell entry and viral immune evasion, including coronaviruses and influenza viruses. Now I'm serving as product development manager in ACRO Biosystems in charge of developing SARS-CoV-2 antigen products for research and diagnosis use, and SARS-CoV-2 kit products for research use. Today, I will start my presentation with the introduction of the outbreak of COVID-19. COVID-19 pandemic is caused by the emerging coronavirus SARS-CoV-2, and it presents a serious global public health emergency. The first cases of COVID-19 were reported from Wuhan, China in December 2019. According to the World Health Organization, the pandemic is affecting 213 countries and territories around the world and 2 international conventions. As of June the 30th, over 10 million confirmed cases of COVID-19 has been reported, and 0.5 million deaths have been caused by this pandemic. In the COVID-19 map offered by Johns Hopkins Coronavirus Resource Center, the red dots represent the confirmed cases of COVID-19, and the higher the red dots, the more confirmed cases in the specific region or country. And you can see that COVID-19 has spread to most parts of the globe. In addition to the health damage caused by the pandemic, COVID-19 has huge impact on our daily life. Now we have got used to wearing protective masks and keeping social distance in the public area. COVID-19 is caused by SARS-CoV-2, which belongs to coronavirus. Now let's take a look at the background of this virus species. Coronavirus was first identified by Dr. Almeida in 1964 in St. Thomas Hospital in London. They are a group of envelope viruses that have a halo or crown-like appearance when viewed under an electron microscope. The genome of coronaviruses are composed of positive sense single-stranded RNA, and their genome size ranges between 27 to 34 bases, which is relatively large for viruses. Coronavirus infect mammals and birds and cause various symptoms such as respiratory tract disease and diarrhea. This virus species can be categorized into 4 genogroups, including alpha, beta, gamma, and delta. And within the beta coronaviruses genogroup, there are 4 lineages, including A, B, C, and D. 7 coronaviruses have been reported to be able to infect humans. Among them, HCOV229E, HCOV-NL63 belong to the alpha genogroup, and HCOV-HKU1, HCOV-OC43 belong to A lineage of beta genogroup. These 4 coronaviruses can only cause common cold. SARS-CoV and SARS-CoV-2 belong to B lineage, and MERS-CoV belong to C lineage of beta genogroup. These 3 coronaviruses have been shown to be lethal. SARS-CoV-2 spike protein is a key transmembrane glycoprotein that forms homotrimers protruding from the virus and mediate host cell attachment and cell entry. It was reported that SARS-CoV-2S and SARS-CoV-S share the same functional host cell receptor, which is angiotensin-2, also ACE2. Spike protein contains 1,273 amino acids. It can be cleaved into S1 and S2 subunits. S1 subunit is responsible for binding to the host cell receptor. It consists of N-terminal domain or NTD and receptor binding domain. S2 subunit mediates fusion of the viral and cellular membranes, which contains fusion peptide FP, peptide repeat 1, HR1, and central helix CH. Currently, spike protein extracellular domain, including both S1 and S2 subunits. S1 subunit, RBD, have been widely used in the serological test development. We'll talk about the application of these antigens in details later. At present, the COVID-19 pandemic within the many countries have not yet reached its peak, and the number of cases and deaths are predicted to rise in the coming weeks and months. Early and accurate diagnosis of COVID-19 is essential, not only to ensure appropriate patient care, but also to facilitate identification of SARS-CoV-2 infected people to control disease spread. The etiological diagnosis of SARS-CoV-2 infection is regarded as gold standard for diagnosis, and nucleic acid testing with real-time PCR has been widely employed. However, high numbers of false negatives have been found in some laboratories, leading to a positive detection rate of RT-PCR, around 50% of suspected clinical and epidemiological COVID-19 cases. In addition, nucleic acid testing is operator restricted, time-consuming, easy to pollution, and expensive. Serological tests can be used to identify whether people have been exposed to a particular pathogen by looking for the presence of antibodies that are produced in response to infections. IgM is first elicited by the immune system in early stage of infection, and then IgG is generated in large quantity after 7 days of symptom onset, which can persist in the blood for several weeks or months. Serological tests offer some advantages over real-time PCR. Firstly, human antibodies are much more stable than viral RNA, so serological specimens are less sensitive to spoilage during collection, transport, and testing than real-time PCR. Secondly, because antibodies are typically uniformly distributed in the blood, serological specimens have much less variations than viral RNA specimens, and they can be easily collected. Thirdly, unlike real-time PCR, serological tests can detect past infection because of the long-term existence of antibodies in blood. Therefore, serological test is an efficient subset of nucleic acid detection, and the demand for serological tests of SARS-CoV-2 is increasing rapidly. Improve the accuracy of clinical diagnosis and better quantitate the number of cases of COVID-19, including those that may be asymptomatic or have recovered. Serological test kits can be 3 types, including colloidal gold immunoassay, chemiluminescence immunoassay, and ELISA. First, let's take a look at the colloidal gold immunoassay. Colloidal gold immunoassay is a kind of lateral flow immunoassay. The test strip is made up of 4 parts: pad, conjugate pad, nitrocellulose membrane, and absorbent pad. Before testing, the gold conjugate antigen is placed on the conjugate pad. IgG secondary antibody and antigen antibody are put on nitrocellulose membrane as test line and control line, respectively. When a drop of contains IgG and IgM is put on the sample pad, the antibodies will move into the direction of absorbent pad. If the serum includes the IgG/IgM against the pathogen, these antibodies would bind the antigen conjugate pad and then interact with the anti-IgG/IgM secondary antibodies at the test line. Hence, at the test line where gold label protein is accumulating, red or pink spots can be seen by the naked eye. The colloidal gold immunoassay strip shown here indicates that the serum contains the antibodies against the SARS-CoV-2, and the patients have been infected by the virus. The colloidal gold immunoassay is easy to use, and it only takes to see the results. Here is the list of colloidal gold immunoassay commercial kits that are approved by FDA. For the chemiluminescence immunoassay, it belongs to labeled antibody technology, and it labels antibodies or antigens with chemiluminescence agents, catalytic luminescence enzyme. When labeled antibodies or antigens are bound with corresponding antigens or antibodies, the luminescent substrates are activated by luminescent enzyme, and then the released visible light or luminescence of fluorescent substances by luminescent spectrophotometer. For ELISA, it employs similar principle as chemiluminescence immunoassays. It uses enzyme-labeled antibody or antigen. The main advantage of chemiluminescence immunoassay and the ELISA is the higher sensitivity to the colloidal gold immunoassay, but they usually need around 1 hour to get the result, and they require instruments or labs to perform the test. After talking about the application of SARS-CoV-2 antigen in serological test development, I'll be able to help fight against COVID-19. First, I would like to offer you a brief introduction of ACRO Biosystems, and we are a leading manufacturer of recombinant proteins and other critical reagents to support the development of target therapeutics. ACRO Biosystems is dedicated to accelerating the process of target therapeutic drug development from the starting point of target identification to the end point of clinical trial. ACRO Biosystems has served over 3,500 customers from around 60 countries and reached out to 20,000 end users from pharmaceutical companies and academic institutes. ACRO has established a collaboration with big pharma such as Pfizer, Novartis, Johnson & Johnson, and well-known academic institutes like NIH. To provide better service to end users, ACRO Biosystems has set up 2 working places at Beijing and Shanghai in China, and 3 offices in Boston, St. Louis, and Newark in the US. With the outbreak of COVID-19 in China, ACRO Biosystems responded quickly and finished the design of SARS-CoV-2 antigen product in Chinese Spring Festival, and immediately started to produce the antigen after getting back to work. Up until now, we have launched 40 SARS-CoV-2 antigen, and another 20 products are under development. The majority of these antigen products are spike ectodomain S1 subunit and RBD. ACRO has started on developing products that can be used in serological test development. We have made following efforts to provide high-quality products to our customers and help fight against COVID-19, including verifying the trimeric state of our spike ECD protein product, the production of biotin protein, optimizing the expression construct for spike protein RBD, developing anti-SARS-CoV-2 antibodies, and producing mutant spike protein RBD. First, let's see how ACRO produced SARS-CoV-2 spike protein in trimeric state. The data of spike protein is required to develop a serological test kit for COVID-19. NIH has published a paper describing the development of an ELISA assay for sero surveys of the SARS-CoV-2 pandemic. The trimeric structures of SARS-CoV-2 spike protein was first verified by analytical size exclusion chromatography and transmission electron microscopy, and then deployed in ELISA kit. Produce trimeric spike protein. We introduced GSAS substitution at the S1-S2 fusion cleavage site to maintain the integrity of the spike protein. In addition, a fold-on trimerization domain was introduced just in the upstream of the predicted transmembrane region to facilitate the trimer formation of spike protein. ACRO Biosystems uses its proprietary HEC293 expression platform to produce recombinant spike trimer protein, and the purity is greater than 90% in SDS-H, and the protein migrates at about 200 kilodaltons under reducing conditions due to cosylation. Since the purity on SDS-H cannot indicate the conformational homogeneity of the protein, we performed stack moss to show that the trimeric S protein is with more than 80% purity, with a molecular weight around 660 kilodaltons. To further characterize the bioactivity of the spike protein, we use surface plasmon resonance to measure the binding ability between spike trimer and its host receptor ACE2. The constant dissociation shows around 35 nanomolar, which is consistent with the binding affinity reported in paper. In addition, we are the first company around the world that provide the negative stain electron micrograph of our spike trimeric protein. The result shows that the well-folded trimeric particles of SARS-CoV-2 S protein are similar in size and appearance to previously characterized SARS-CoV-2 trimers. We also perform stack moss to compare our SARS-CoV-2 spike protein product with the protein offered by other vendors. As you can see from this slide, in contrast to ACRO's trimeric spike protein product, the conformational purity of 97.59%, the spike protein offered by other vendors are in high polymer form with molecular weight around 4,500 kilodaltons. In addition to unconjugated antigen product used in serological test kits, the Biotinylated SARS-CoV-2 recombinant antigen is also employed in commercial chemiluminescence immunoassay products. On May the 29th, Siemens Healthineers Diagnostics receives FDA emergency use authorization for its SARS-CoV-2 total antibody test. The total antibody test has demonstrated 100% sensitivity and 99.8% specificity in identifying SARS-CoV-2 antibodies. The antigen used in the antibody test is biotinylated SARS-CoV-2 recombinant antigen, which bind the streptavidin-coated microparticles and capture anti-SARS-CoV-2 antibodies in the specimen. ACRO Biosystem has developed an exclusive collection of pre-labeled and experimentally validated biotinylated SARS-CoV-2 spike proteins. These products are produced using the AVI tag technology, which introduces a unique 15 amino acid peptide AVI tag into the recombinant protein during expression vector construction. The single lysine residue in the AVI tag is enzymatically biotinylated by the E. coli biotinylase A. In this way, the biotinylation only happens on the single point lysine residue on AVI tag. The protein orientation is uniform when immobilized on an streptavidin-coated surface. One of our customer drugs and diagnosis for tropical disease has commented after using our biotinylated spike proteins. The AVI tag biotinylation technology from ACRO Biosystem is a great fit for our LFA platform. Recently, a paper published by NIH has shown that different RBD constructs have a distinct signal when used in developing serological ELISA assay. The two RBD constructs from Kramer Lab of Mount Sinai and Reagan Institute were compared, and the construct from Reagan Institute displays stronger signal with a recombinant RBD antibody. ACRO also got feedback from our customers that the appropriate RBD construct can determine the sensitivity and the specificity of ELISA kit. We are currently working on optimizing the RBD construct to increase the sensitivity while maintaining the specificity of the kit product, and this product has been at the end of June. ACRO noticed that several RBD mutants have been reported in of SARS-CoV-2, and we generate the mutant protein products to characterize the difference between wild type and mutant RBDs. ELISA and SPR results have shown that mutants shown in this table combine ACE2 with 2 to 6-fold higher affinity compared with the wild type RBD. These mutant RBDs can be used neutralizing antibody of anti-SARS-CoV-2 antibodies to exclude the possibility that the mutation in RBD makes the protein unrecognizable for the antibody. We tested binding ability between our anti-SARS-CoV-2 antibodies and the RBD mutants. These antibodies all show similar binding affinity with RBD mutants and wild-type RBD in SPR assay. In addition, the antibodies can inhibit the interaction between RBD and ACE2. These studies indicate that RBD mutants developed by ACRO are useful research to verify the broad spectrum neutralizing antibodies against different strains of SARS-CoV-2. In addition to offer SARS-CoV antigen products, ACRO collaborated with its partner to develop anti-SARS-CoV-2 antibodies as well. ACRO Biosystem has developed a variety of mouse and chimeric anti-SARS-CoV-2 monoclonal antibodies and mapped the epitope of these antibodies. Anti-SARS-CoV-2 S1 and N protein antibody pair has been launched for serological assay development. The sensitivity of detecting nuclear capsid protein by anti-SARS-CoV-2 nuclear capsid antibody pair in our ELISA assay is 20.5 pg per milliliter. ACRO Biosystem has developed SARS-CoV-2 antigen and antibody products specifically designed and optimized for serological test kits. Spike-derived proteins are produced from HEC293 cells, while nuclear capsid protein are either yielded from HEC293 cells or eclipse cells. The antigens are expressed with a variety of tags, including HES, human FC, mouse FC, and HES AVI tag, which could meet the needs of colloidal gold immunoassay, chemiluminescence immunoassay, and ELISA kit development. Here is the list of our products specifically designed for serological test kits, and you can contact us if you want to know more about these products. Now, this will be the last slide of this presentation. Thanks everyone for attending today's webinar. We're doing a raffle for everyone. You will have the chance to win our adorable llama. They're very popular among our customers. If you have not already entered this raffle, you can go to our website and enter it. We will contact you with business days if you win the llama. Okay, that's all for my presentation. And now I would like to answer the questions from the audience. Thank you.
Now I would like to pass the presenter ball over to our final presenter, Dr. Stephen Rawlings. Welcome, Dr. Rawlings. The presenter ball is yours.
Thank you. Thank you, Elizabeth, and everyone for attending, and to my prior presenters. I hope I don't have too much that's overlapping with their intro slides. Oops. So I just want to say I'm a clinical infectious disease fellow at UC San Diego, and I've been working, I trained predominantly in HIV, virology, and immunology, but with the emergence of the COVID-19 pandemic, have shifted gears slightly along with my mentor, Davey Smith, and a number of others. I have no disclosures. I'm exclusively paid by hospital dollars and NIH training grants. And I don't need to spend too much time on this, but just a brief reminder. So we use serologies in general to assess evidence of a prior infection or immunization for pathogens. We've talked a little bit about point of care tests. These are ideal for a number of purposes. They can have minimal lab supply and support requirements. They can be done in the field or the parking lot at Qualcomm Stadium, where people can drive up and get their finger pricked. They have usual sort of waivers for CLIA and the stricter requirements for a lab test. But at the same point, I'll point out that other serologies are being done here at UCSD and other institutions or through methods that are not just lateral flow assays, but other platforms as well. And we obviously are all interested in a COVID-19 serology for a number of reasons. We know that this infection has asymptomatic presentation. If we could understand how many people truly have been asymptomatically infected, this could give us an idea of the true attack rate. Perhaps we're missing some spread of cases by not understanding who's really been infected. At the same time, if we knew that there were more people infected, but the same number of deaths, that could actually decrease the case fatality rate and give us a better understanding of what the death rate from this, not that we want to know anymore, but necessarily, but it's still an important marker of this disease and pandemic. There were also, just to point out, I mean, sort of on a personal level, people in February and March had illness that was either mild or not severe enough to get tested when we had a scarcity of PCR tests. And those individuals would like to know if what they experienced was truly COVID at the time. And we've talked a little bit about this in the prior presentation, but as we know, SARS-CoV-2 is a novel coronavirus that's part of this larger beta coronavirus family, and then there are alpha coronaviruses as well. Sequence homology suggests it's most closely related to the etiologic agent of the 2003 SARS outbreak, which is still gets called SARS-CoV. Maybe someday we'll start calling it SARS-CoV-1. The next closest relative is MERS, which fortunately didn't make it much farther than a few infections and sadly deaths many years ago. And then circulating human coronaviruses that cause sort of seasonal flu-like illness, beta coronavirus OC43 and HKU1 are the next most similar, followed by the alpha coronaviruses that also circulate and cause seasonal flu-like symptoms, N63 and 229E. And so some people, when they get tested for flu in a normal year, and the doctor says, well, you have the flu, but it didn't come up positive on the flu test, many of those cases are just these coronaviruses. And those individuals may have antibodies against the coronavirus afterwards, although we do know that some coronaviruses don't elicit very strong antibodies or the antibodies can be cross-reactive. And so that poses issues when trying to assess individuals for evidence of infection by this novel SARS-CoV-2 coronavirus. And so this is just a slide from a preprint a couple of weeks ago that had an interesting experiment. So they had blood from 3 different groups. The black circles are archived blood samples from before 2019, so before SARS-CoV-2 was circulating and could possibly have elicited any response. The pink circles are from participants in New York and New Jersey who had confirmed and symptomatic SARS-CoV-2 infection confirmed by PCR. The teal, sort of green-blue circles there were from people who were asymptomatic and did not have a positive SARS-CoV-2. And so when those plasma and serums were placed in an ELISA with various spike proteins, this first is SARS-CoV-2, then MERS, SARS-1, and then 2 of the circulating coronaviruses, you can see They didn't find any cross-reactivity against the SARS-2 spike from people who had never seen it, obviously, which makes sense. And then there was a fair amount of reactivity in people who had confirmed symptomatic infection, although a couple of people did not seem to. And then also asymptomatic people, participants in the study, did occasionally react against SARS-CoV-2. And I'm just seeing some of these comments. Sorry, I will try to get to these, but oops. So, and then there's a lot of antibody against the circulating strains, consistent with the fact that these infect people sort of variously throughout the seasons. So what did we do at UCSD? So in February of 2020, our group recognized that there would be a need to collect and bank samples from patients infected with SARS-CoV-2 for various research studies. And on February 12th, our IRB approved a protocol to collect a number of biologic fluids, so saliva, sputum, urine, stool, semen, breast milk, and importantly, blood from participants with known or suspected infections. And we have the ability to collect while people are acutely infected, as well as in the convalescent phase on sort of a monthly basis. Since February 12th, we've enrolled over 140 participants, roughly breaking down to about 25% acutely infected and getting samples from them while they're infected. 25% of participants were exposed at some level, either to a loved one or a workplace exposure. Many of them ended up not converting, but they are in the study nonetheless as some useful controls. And we have about 50% of the participants, more than 70 samples, 70 participating individuals who have given convalescent blood and saliva in some cases. And they had either confirmed or in a few cases, because it was March and some people weren't getting tests, a very strong suspected infection that we're able to sort of use to validate some of our assays. And we have published already, just to sort of highlight some of these, looking at the T-cell responses, isolating neutralizing antibodies, and also looking at the immunodominance of different proteins from coronavirus. And I'll summarize our findings here. We did find in at least an initial segment of the cohort that 100% of participants had CD4 T cells that could recognize some part of SARS-CoV-2 antigen presented to them. 70% of them had CD8 T cells that recognized the virus. There was a fair amount of cross-reactivity, though, because these same peptide pools were recognized by 30% of donors from before 2019. So we're still working on teasing out which parts of the virus is most specific to SARS-CoV-2 memory. We also found, again, in just a subset of the participants from the early part of the study, but about 15% had strongly potent neutralizing antibodies. That's not to say that 85% of participants did not have neutralizing antibodies. We were looking for the strongest ones. But because we take whole blood, we were able to sort out B cells, clone them, synthesize their neutralizing antibodies, and show in a hamster model of SARS-CoV-2 infection that infection could be prevented in sort of a passive transfer immunity experiment. And that one other paper also seemed to suggest that the nucleocapsid was a little bit more antigenic than spike, but there's a lot of ink spent on these that I won't get into as we tease apart more about the immunodominance of various epitopes in the virus. As you can imagine, and relevant to this presentations, a number of companies approached us to use our clinical samples on their lateral flow serology assays. And not to throw stones, but there was some initial performance that wasn't always great, but this has improved dramatically. I'll just sort of highlight, things have been in the media, and it's important that as scientists and clinical researchers, we try to make sure that good antibody tests are making it to market. So the most recent test that we have worked on, and that is being, the company is submitting data to the FDA for approval, has characteristics close to 98.4% sensitivity, 98.9 specificities. One of the things that I know, without knowing a ton about the dynamics of that test, that when 2 viral antigens were used instead of 1. That improved the sensitivity a lot in the immobilized platform. We do have a number of participants, just a handful in our study, who have confirmed swab positivity for SARS coronavirus, but are persistently negative on a variety of antibody platforms. What we're still trying to tease apart, and it's going to take some time, is understanding, do they have a rare antibody that does suggest that they had B-cell immunity or memory against the virus, but it's a very rare antigen that's simply not being detected on the majority of commercial tests, versus do they actually lack any antibodies specific to SARS-CoV-2, and perhaps their immunologic memory is restricted to the T-cell compartment. Here's just a sampling of some of the tests that we've done. So here's 2 different commercial tests with the same 5 different donors. These were all interpreted as positive. You can see, as we kind of saw in some of the other slides, that lines can sometimes vary in intensity. We're not clear that anything really means much difference clinically if somebody has a very strong line versus weak in these particular participants. So our future directions that we'd like to move in include being able to test saliva for the evidence of antibodies. This would obviously be easier, less invasive, if people could just spit into a tube at that line, waiting in line for a test at Qualcomm Stadium here. If you could make it, it would be amazing to get a combined antibody-antigen saliva test, so something that could simultaneously look for acute and convalescent infection, or rather acute versus convalescent. So maybe your antibodies are negative, but it turns out you have antigen that's there, or you are symptomatic, the antigen's negative, but the antibody's positive. Perhaps you have an early or medium length recency of infection. We are longitudinally sampling participants in our study to understand the durability of antibody response. Is it something that lasts months, years? We're not sure. So this is a new virus. The oldest infection was 7 months ago. So we need to understand how long immunologic memory will last for these participants. And then it would be interesting to understand if there were a particular epitope that's required for neutralization, or rather if there's some antigen that is specifically a weak spot in the virus, and antibodies against that is quickly neutralizing. If that could be immobilized on a point of care lateral flow test strip, we could give particular individuals information about their ability to neutralize the virus. And just to sort of tease, here are some saliva versus plasma comparisons. So 2 different individuals on the left, no detectable antigen in saliva. This is just basically putting saliva on what is supposed to be a plasma or whole blood test, so not completely using it for its intended purpose, although their plasma was clearly positive. Interestingly, a different participant had very weak plasma line, but there is a detectable line in their oral secretions. Not clear if this is some variance in their expression of antibody in different compartments in the body or what, but this is an area that we're actively pursuing. And I just want to acknowledge my mentor, Davey Smith, and a number of the other collaborators for our clinical study, the participants and their families, the staff here at the AVRC who've helped collect blood, and we have a lot of support at our San Diego Center for AIDS Research. And I saw lots of pop-ups, so I will stop sharing and take questions.
All right, we have a couple for you. The first one from Lourdes: how is the cross-reactivity of the S1 and RBD antigens in the assays between SARS-CoV-2 and other coronaviruses?
And I saw that pop up. I'm afraid I do not have the expertise to answer that. I'm not intimately involved in using the different antigens in these assays. I've mostly been characterizing the participants. So I think that somebody would know this. If you want to send me an email, I'll try to figure out the right person to forward it to. I'm sorry, I don't have the answer to that. And similarly from Lourdes, the N protein antigen to differentiate from other coronaviruses. Again, I think this has been worked out to try to reduce that cross-reactivity, and for the most part, we're not seeing a lot of cross-reactivity in the later generations of tests. And to try to answer also Israel's question from before, one of the things I do think is powerful is using archived plasma or blood samples, because we essentially know when this virus crossed into humans was fall of 2019. So if you have blood from 2018 or before, it's very useful to put it on and help test specificity. Okay.
You have one there also. Did you measure antibody titers?
Yes, so we do measure antibody titers. We have not yet collated all the data to correlate to, say, intensity on the bands on different tests, but we do separately. So our blood draws are almost a half a pint of blood, and it's used by a number of different investigators, and so a number of different ELISAs are done as well, and we are in the process of still collating all that data.
From Susu, did you measure the antibody titers, including IgA?
Right, so I don't think we are yet on a wide scale measuring IgA antibody titers. We do have a couple of test kits that have been sent to us from companies that are using IgA as part of like a combined IgA, IgM, IgG test. And that's had various performance, but it is being looked at. And I know other groups have certainly found IgA reactive specific to coronavirus. That's also, obviously, we would want to use that in the saliva test because classically, at least, IgA is much higher levels in saliva.
Excellent. We have time for just one more question. There are a whole bunch more, but I'll be sending those along to you via email.
All right, sure.
This one is from Abhishek. Do you think lysis of virus from Silva might provide you with better LFT?
I don't know what Silva is. So, and I'm sorry, I don't fully understand.
Sorry, he meant saliva. Thank you for clarifying, Abhishek.
Lysis of saliva would give us better results. So, I don't know for sure. I think we need to figure out the correct handling of the specimen. So part of this is obviously we have an eye towards making this a very easy point of care rapid test. And so whatever lysis or manipulation needs to be done should be done ideally at the site with sort of minimal handling. But generally, we consider antibodies to be freely secreted into saliva. So I don't know that we would necessarily need to lyse anything, but I'd be happy to entertain, thoughts on that.
Okay, and time for just one last test, or one last question. Which antigen should be targeted while developing an antigen rapid test for COVID-19?
Yes, I'm not totally sure. I think it almost wouldn't matter as long as you just pick an antigen that's gonna, be present in. Right. I mean, we were just looking for whole virus. If you want antigen tests, you just want to know that virus is present. If it's slightly decayed virus, that's fine too. So maybe you want the most stable antigen, but that's something that I think we should try to sort out. But yeah, I don't have an answer yet.
All right, thank you. We do have quite a few more questions. I'll compile those and send them along to you. So we've run out of time. But I would very much like to thank you, Dr. Rawlings, for your presentation, our presenters, Dr. Yinan Jiang and Dr. Marco Biamonte for their presentations, and for the folks at ACRO Biosystems for sponsoring today's event. And most of all, I'd like to thank those of you who came and spent this time with us. We know that it's a crazy time, and you have a lot of choices for how to spend your time, and we're very grateful you chose to spend it with us. So on behalf of Cambridge Health Tech Institute's Breakout COVID-19 webinar series, thank you all so very much, and have a great rest of your day. Bye-bye.